Hello, everyone. I'm Janet Rossant. I'm President and Scientific Director of the Gairdner Foundation, and it's my pleasure to welcome you to Day 2 of the 2020 Canada Gairdner Laureate Lectures. I'm speaking to you from Toronto, Ontario, Canada, where we have the pleasure every year of hosting the annual Laureate Lectures. This year, of course, our lectures are not physically in Toronto, they're completely virtual, featuring our Canada Gairdner Award laureates joining us from their homes and offices around the globe. Yesterday, our laureate lectures featured Dr. Rolf Kemmler in Freiburg, Germany, Dr. Masatoshi Takeichi by video in Kobe, Japan, Dr. Mina Bissell in Berkeley, California, and Dr. Elaine Fuchs from New York City. Today, we've got the pleasure of hearing from Dr. Guy Rouleau in Montreal, Canada, Drs. Quarraisha and Salim Abdool Karim in Durban, South Africa, and Dr. Roel Nusse in Palo Alto, California. Today's virtual event is being streamed all over the world thanks to the generous support of our partners, the National Research Council and the University of Toronto. I encourage all of you watching to follow us on Twitter, Instagram, and Facebook at Gairdner Awards and share your favorite parts of today using the hashtag Gairdner2020. Now, just a little bit about the Gairdner Awards. Annually, we give 7 Gairdner Awards given out, 5 Canada Gairdner International Awards for outstanding biomedical research with impact on human health, 1 John Dirks Canada Gairdner Global Health Award, specifically for research with impact on global health issues, and 1 Canada Gairdner Wightman Award, reserved for Canadian scientists showcasing outstanding science and outstanding leadership. So today we're going to begin with this year's Canada Gairdner Wightman awardee, Dr. Guy Rouleau.
The major challenge that has been the focus of my work has been understanding the basic mechanisms of diseases of the brain. It was important to focus on understanding mechanisms of brain diseases because we understand so little about the brain that we have a lot of trouble actually developing new treatments. And so understanding the mechanisms of brain diseases makes finding new treatments easier. I'm being recognized for 2 bodies of work. The first is the understanding and identification of genetic factors that predispose to diseases of the brain. And these can be either disease-causing genes or it can be genetic mechanisms. An example of a genetic mechanism is exploring de novo mutations as a cause of neurodevelopmental diseases like autism and schizophrenia. I'm also being recognized for work that I've done to explore a new way of doing research, which is essentially one where one breaks down barriers to collaboration so that there's more sharing of information. This is called open science. The goal of open science is to accelerate discovery. And we need to accelerate discovery to be able to find new cures and to be able to help people. What I would like most would be that my research would lead to the development of new treatments for some of the common brain diseases. And this is starting to happen. There is some disease mechanism-focused treatments that are starting to be explored, but I think that this is ultimately going to be the largest impact, is going to be all the different treatments, new treatments that are going to come from what I've done. This prize is really very important for me because it recognizes 2 things. It recognizes my lifelong work identifying genes and mechanisms for brain diseases. And it also recognizes the work that I've been doing to pioneer open science. When I got the call from Dr. Rossant informing me that I had won the prize, I was really overjoyed. I was very excited. I knew exactly what this prize is, and it is by far the greatest recognition of my work that I could possibly have. Good afternoon or good morning. I'm very happy to be here today to be able to speak to you about my work. As was mentioned in the video, I'm being recognized for my work on neurological diseases and for open science. And I'm going to give a small overview of the kind of work that I've done with some concrete examples of the thinking and some results. And so I'm going to basically talk to you about from neurogenetics to open science. I have a disclaimer. I have no conflicts of interest or financial interest to disclose. The people who are in my lab require that I put another disclaimer, which in fact is I done none of the work that I'm going to be talking about. So, amyotrophic lateral sclerosis, the first disease I'm going to talk about. This is a neurodegenerative disease that involves amyotrophy, which is basically losing muscle mass. And you can see in the middle here is muscle fibers that have been denervated and have atrophied. So atrophy of muscle. And one gets motor neuron death. And these motor neurons are in the anterior horn of the spinal cord and in parts of the brainstem. And one gets what's called lateral sclerosis. So this is a transversal cut of a spinal cord. And what you see in white here on both sides are the corticospinal tracts that are basically degenerating. And that leads to sclerosis of the spinal cord, plus there's loss of mass in the anterior horn. So that's where the name comes from, amyotrophy and lateral sclerosis. My work is focused on the familial forms of ALS, and familial ALS basically constitutes about 10% of cases of ALS. It's primarily autosomal dominant, but there are some recessive families and some small families that probably have incompletely penetrant genes which are predisposing to them. The disease is clinically indistinguishable from sporadic ALS, except the age of onset is a bit younger, and the sex distribution is even, equal male and female in familial, but there's an excess of male who get ALS. So the story of familial ALS began in this journal in 1880, Archives of Medicine, in a paper that was published by Sir William Osler, who was at McGill in Montreal at that time. And he described the first family with familial amyotrophic lateral sclerosis. So on hereditary in progressive muscular atrophy as illustrated in the FAR family of Vermont. So in this paper, he describes this family. And in this family, you can see bolded are the individuals who are affected. So one sees pretty classic autosomal dominant inheritance. One has, if one thinks of ethics, one normally wouldn't put the name of the family and all the family members in a publication, but I guess in 1880 it was a different world. And in fact, one might not put certain information like this person died a drinker, definitely not something that one would include in publications today. So he described the first family. ALS is a very bad disease in that this is basically a survival curve, the years at the bottom, and basically 50% of people have died within 2 years. There are some people who are more long-term survivors, 8 years, even some up to 10, 12, 14 years, but those are very, very rare. So this is a relatively rapidly progressive, almost uniformly fatal disease for which there was no disease-modifying treatment. The story of my involvement in ALS actually began in 1986, but the most important event was the identification of the SOD1 gene, which was the first gene identified that would cause familial ALS. It accounts for about 15% of the familial forms and 1 to 2% of all ALS cases. There are many different mutations that you can get in this gene that can cause the disease. In fact, the gene has 152 amino acids, and so 168 mutations, over 168 mutations identified to date. So you can have even different mutations of the same amino acid that would predispose to the disease. They're all dominant with the exception of a few mutations like D90A in particular and D96N. There is some genotype-phenotype correlation, so some mutations affect survival time. So there are what's called the rapid progressors. The A4V mutation is one of the most common rapid progressive diseases. You should know that this is a mutation that is in the Vermont family described in 1880 by Dr. Osler. There are also other alleles that lead to slow progression. Some have early disease onset, some have later disease onset. A lot of work has been done. There's been thousands of papers written on SOD1, and really it was the first gene identified and the only gene for quite a long time, and led to a lot of improvement in our understanding of ALS. The next gene that was identified was TDP43, also called TARB-D2. DP, and this was the first paper was by Newman et al., who basically took brains of patients with frontotemporal dementia, isolated the inclusions, isolated the protein, the insoluble protein in the inclusion, sequenced it, and found that the protein was TDP43. And so there was these inclusions had many proteins, but one of the important proteins and abundant proteins was TDP43. When we saw this, we decided that this would be a good candidate to look for ALS, because there had been many reports of ALS and FTD, frontotemporal dementia, coexisting in the same families and even in the same individuals. And so we selected 200 patients with ALS, some with familial ALS, some with sporadic ALS, and we sequenced this gene, TARB-BP. And what we found was 9 mutations out of the 200 patients, 3 familial ALS, 6 sporadic ALS. 1 mutation showed segregation in the family. The other families were small and didn't have more than 1 affected individual, so we couldn't look at segregation. And we found only 1 missense mutation in the controls. And so this is a summary of all the different mutations, and what you can see is that first of all, nobody has frontotemporal dementia in this family. They all have pretty pure ALS. There are a number of individuals who have familial ALS. There are a couple of recurrent mutations. So this was certainly a strong indication that TDP43 or TARB-BP could predispose to ALS. At the same time, another group found mutations in this gene in a couple of families with familial ALS. And since then, many people have confirmed that this is an important gene in familial ALS. When people started doing TDP43 immunohistochemistry in spinal cords and brains of patients with ALS and frontotemporal dementia, it was found that the vast majority of cases of ALS, whether sporadic or familial, had mutations had TDP43 in their inclusions. So this seems to be, if it's not a common final pathway, it's certainly an important pathway for the development of ALS. The only individuals who have ALS who do not have TDP43 inclusions are the SOD1 families and the FUS families. And this is just a diagram showing that the mutations really cluster in this part of the protein, which is a prion-like domain. It's not a prion, obviously, but it's a prion-like domain that has properties similar to prion. And this contributed to the hypothesis that ALS is caused by a prion-like spread of misfolded protein, of which TDP43 is one very important component and a candidate for this spread. This hypothesis still remains a hypothesis, but the amount of evidence mounting indicating that this is a mechanism in ALS is increasing. And this is just a diagram showing that over the years, more and more genes are being identified, a bigger and bigger fraction of the number of cases are being explained, and there are many genes here that are indicated to predispose to ALS. Most important, SOD1, TDP43, FUS, which I'm not going to talk about, and C9orf72, which I'm not going to talk about as well. The others some of them are definitely disease predisposing. Many others might be risk factors, and others may be false positives, so there's still a lot of work to be done to be able to identify these. This is the fraction of cases explained by these genes in sporadic ALS, and you see we're not quite at 25% now, indicating that there remain probably very important genetic factors to discover and also environmental factors. I just want to go around. This is not my work. This is from a clinical trial of tofersen, which is an antisense for SOD1. And this graph here basically shows that with a higher and higher dose of intrathecal antisense, one gets a greater and greater reduction in the levels of SOD1 in the CSF. But I think what's important is this graph, which basically shows, and I'd like you to look mostly at the second part. So they separated all the ALS cases treated and the fast progressive subgroup. And if you look at the fast progressive subgroup, this is the normal course of ALS with a steady deterioration. And these are individuals who are being treated with this antisense, and you see that their disease is essentially halted. And so what we're seeing here is the first effective disease-modifying treatment in ALS. And it is a treatment which is right now focusing on rapid progressing subgroup, but also on other individuals with SOD1 mutations, and is currently being tried in all cases, including sporadic ALS, because there is one hypothesis that sporadic ALS, the final common pathway is SOD1, but that remains to be proven. I should mention that the Farr family, the Vermont family described in 1880, has one of these rapid progressive mutations, and there are a number of these patients from this family, descendants from that family, who are included in this study, who are actually being treated at the clinical research unit here in Montreal at the Neuro. So a nice circle from identifying the family, and 140 years later, finding a disease-specific treatment for this family. So I'm going to stop about ALS. I'm going to talk about neurodevelopmental disorders. And, you know, there are many, many neurodevelopmental disorders. I'm going to focus on two, which are really quite common, schizophrenia and autism. So schizophrenia, major mental disorder, affects about 1% of the population, wide spectrum of symptoms, but mostly cognitive, behavioral, and emotional problems. The genetics, twin studies, there are over 30 twin studies, modern twin studies. They show heritability quite high of 82 to 84%. Linkage studies, though, and GWAS studies have really failed to explain the bulk of this heritability. Only a few genes had been convincingly linked to schizophrenia. CNV studies did suggest a role for these genes in schizophrenia, including de novo mutations. And so little of the heritability has been explained. Autism, similar story. This is a disease, younger onset, with problems with social interaction, communication skills, language, and repetitive and restricted patterns of behavior. The age of onset is younger, and the incidence or the prevalence is kind of a moving target, but it's now thought to be quite high, around 1% of the population. Autism genetics, same story as for schizophrenia. Twin and family studies show very high heritability. Linkage and GWAS studies do not explain the bulk of the heritability. A number of genes have been identified, and if one goes back to when these studies were done, there were very few genes who had been identified, and they explain only a small number of cases. CNV studies suggested a role for these in schizophrenia, including de novo mutations, so little of the heritability was explained. So when we started thinking about this some years ago, we wondered, you know, where is the missing heritability? And you have to remember that heritability is measured using the monozygotic and dizygotic twin concordance. And so the classic explanation is that if there's a very large discordance between the rates in monozygotics, the concordant rates in monozygotic and dizygotic twins, suggests a polygenic inheritance. So if there are 10 genes that predispose to disease, the monozygotics have all the same genes, so they have a higher likelihood of having the disease. But dizygotic twins would only share maybe half of these, and so less would share the disease. But is there another possible explanation? That's what we were wondering at the time. So I think maybe, yeah, so where's the missing heritability? In fact, de novo mutations, we thought, could explain de novo mutations of many different genes could establish new families with hereditary diseases. And de novo mutations, you know, we know cause many different diseases, including, for example, neurofibromatosis. 1 out of 6,000 live births is a de novo mutation. There are thousands of brain genes, and there must be de novo mutations in these genes. We wondered what would be the phenotype of a gene that would inactivate any of these thousands of brain genes. We thought that many should basically affect neurodevelopment. And so de novo mutations, so in any of these genes, should be relatively common occurrence because there are thousands of genes and de novo mutations are not that uncommon. So we thought that de novo mutations also could explain the large monozygotic dizygotic twin concordance and explain part of the missing heritability. This concept is actually quite simple. Monozygotic twins, if there's a de novo mutation, will share the de novo mutations. But in dizygotic twins, of course, if one has a de novo mutation, the other one will not have the same de novo mutation. So that's a simple explanation. So our hypothesis for the genetic architecture of schizophrenia, autism, and somebody working with us in parallel is working on intellectual disability, was multiple rare variants that are generated by de novo mutations. And we thought this fit the complexity of the brain. So this hypothesis has two predictions before we even test the hypothesis. One is there should be a reduced reproductive fitness. So rates of reproduction are in fact reduced in schizophrenia and autism. And so given that, natural selection should reduce the number of mutant alleles in the population because these are severe diseases with reduced reproductive fitness. However, schizophrenia and autism are maintained at high prevalence worldwide. And so there are two possible explanations. One is that there's a strong positive selection for some of the deleterious genes. The other one is that new alleles are continuously generated through de novo mutations, replacing the alleles that are being eliminated through selection. So, and the other thing is that the relatively uniform incidence of autism and schizophrenia across the world and many different environments kind of argues against a positive selection. You would think positive selection might be different in different parts of the world. It doesn't have to be, but you think that would frequently be the case. So this did support the de novo mutation hypothesis. The other is the prediction is an effect of paternal age. We know that the maternal to female ratio of de novo mutations is 4 to 6 to 1. The mutation rate increases with paternal age. It doubles over about 20 years. So it predicts that de novo mutations should more frequently come from males, particularly older males. And this well documented in diseases like Rett syndrome and NF1, where a lot of the cases are de novo mutations. And clinical studies do show an association between increased paternal age and schizophrenia and autism. It does support the de novo hypothesis. So we put together a project which was called Synapse the Disease Project. This is pre-next-gen sequencing. And so here, our hypothesis is the one that I said, and we wanted to sequence genes of schizophrenia, autism, and intellectual disability as well. And we wanted to focus on synaptic genes because we thought that's where there might be the best bang for the buck. So basically, this project was identify the genes in the synapse and screen these in a large number of cases with autism, schizophrenia, or intellectual disability, and then validate the alleles using various models. And I'll show examples of that. So some results. Firstly, when we got to the 402 gene mark, we stopped and looked at what we had, and we had 15 de novo mutations that had been identified. Interestingly, the ratio of nonsense to missense de novo mutations was 2 to 5. Well, the model, the neutral model predicts it should be 1 to 20. And in fact, there's a significant excess of nonsense mutations in the de novo mutations identified in cases with autism or schizophrenia. So that suggests de novo mutations could be involved. Second was to look at the number of de novo mutations. One would have expected that the frequency or the amount of de novo mutations you would have would be based on the de novo mutation rate, which is 1.8 times 10 to the minus 8. But in fact, what we found was 6.5 times 10 to the minus 8. So there were more de novo mutations in cases than in controls, and that too was statistically significant. So we started drilling down on some of these genes. This is one example. We did this for many, many, many genes. But this is one example, de novo mutation in IL1-RAPL1 in an autism spectrum disorder patient. This is the pedigree. The parents are normal. The child has a 7 base pair deletion that leads to a frame-shifted protein. Here's the smaller protein. This is where the deletion occurs. This is an X-linked gene, so this is why the father only has one allele, and the daughter has a wild type and a mutant allele, the wild type and the mutant allele. So this is just an example of the kind of study we did. This is in cultured neurons, mouse neurons, and what we do is we stain for IL1-RAPL1, and we see that IL-1, RAPL1 is in the dendrites where it should be. We knock it down with microRNA, it disappears. We knock down the gene from the RNA from the patient, and we rescue with wild type messenger RNA that is not susceptible to degradation by the miRNA, and you see that you have restoration of IL-1, RAPL1 in the dendrites. But if you try to replace, correct the abnormality or the knockdown with the mutant protein, you can't, indicating, not surprisingly, that this protein is non-functional. We also looked, for example, here, this is two families that had de novo mutations in the SHANK3 gene. These are both schizophrenia families. Here it was three brothers, actually, and we could show that the father was a germline mosaic for this mutation, which is a protein truncating mutation. And we found this individual who has a missense variant. This is a work using zebrafish. And normally these zebrafish are, normal, have good swimming. If you knock down the gene, they have a severe phenotype. If you knock down the gene and rescue with the wild type, they have largely a rescue of the phenotype. If you knock down and try to rescue with the truncated protein, this protein, then the rescue is not, there's very little rescue. And there is quite adequate rescue with this missense variant. So we don't know if the missense variant is a de novo mutation that has no effect or it has a mild effect. We have not been able to determine that. So SHANK3 in schizophrenia. I should mention that SHANK3 has been implicated in schizophrenia and in autism, and some people have looked at the function of these proteins in mice at these different mutations and find different phenotypes. So our outcomes, basically, we provided support for the hypothesis that schizophrenia, autism, de novo mutations are an important cause. So when we completed that, next-gen sequencing had arrived, so we said, let's reproduce our own study using next-gen sequencing. And this is the results. We found, again, I think it's 15 mutations. These are the protein truncating mutations in individuals with schizophrenia. These are all de novo mutations found by next-gen sequencing. And we found, in fact, an excess of de novo mutations. You would predict there should be 6.87 de novo mutations. We found 15. This is an excess of de novo mutations in the individuals with schizophrenia. The type of mutation, again, the same story, an excess of nonsense mutations, 4 of 15 as opposed to 1 of 20. And so there's an excess of protein truncating mutations in the schizophrenia cases. So our conclusion is our second study replicated the first study. In fact, the second study was the first replication of the first study. I should say that when we tried to publish the first study, it took us 2 years, and it was very, very difficult to publish because it was not considered to make very much sense. Now, though, it's been really quite established that de novo mutations have an important role, particularly in autism, but also in some fraction of cases of schizophrenia. So overall conclusions: hundreds, maybe thousands of genes predisposed to autism, schizophrenia. De novo mutations play an important role. So last subject, open science. So essentially, I'm a neurologist, and when I see patients, I started seeing patients in 1983 when I was a resident, and I'm seeing patients now in 2020. We really have, for most diseases, no better treatments. There are some exceptions, like for multiple sclerosis and stroke, but for the vast majority of patients, no new treatments, no disease-modifying treatments. Things are going very slowly, and we needed to accelerate the pace. It's going slowly because the brain is complicated. It's not because there isn't a lot of smart people doing great work. It's just that the brain is very complicated. So we thought that open science could be a game changer. It would accelerate discovery. It would help with understanding the brain, which is complex. And basically, we wanted to break down barriers for collaboration, allow reproducibility of data, wanted openness, transparency, and a collaborative approach. And essentially, we thought also it was a better use of public money. So the institution, the Neuro, we thought that this fit very well for what it is that we wanted to do, which is understand the brain, find cures, and effectively treat people with neurological disorders. So we thought that open science would be something that would allow us to do this better. So, but we didn't kind of just wake up one morning and say, let's be open. We said, okay, let's do a consultation process, which took 18 months, basically mapping the existing open science activities, consulting with faculty, staff, and students, defining open science, did a social science study to look at the barriers to open science, define the guiding principles, and so on and so forth, and basically convince the university that we should and could adopt an open science philosophy and approach. Mr. Tannenbaum really liked the idea, and we created the Tannenbaum Open Science Institute, and we had the official opening. I'm having some difficulty. Official opening in 2016. This is the principal, my boss, and this is the Prime Minister of Canada. I'm sure you all know. And this is Mr. Tannenbaum. So he donated money, and the money was donated for two missions. One was to implement open science practices at the Neuro, and the other was to encourage other institutions to embark, inspired by the Neuro's experiment. So two different missions. So for the first mission, implementing open science at the Neuro, these are the open science principles. They're all on our website. Release of information and data and biomaterials, no later than publication. Convince others to do the same in our collaborations. Informed consent, privacy and rights respected. No restrictive intellectual property, and autonomy. There are many components to open science. I'm going to talk about two, the open biorepository and the early drug discovery, but there's also open data and IP compatible with open science and open access. This is a bit complicated, but this is our clinical information, biological materials, biosamples, imaging data, and genetic data is not shown here, repository, including longitudinal information on patients, because we are a hospital as well as a research institute, and that we would make this all publicly available. This is going to go online, live, on November 11th, which is when we have our Open Science Symposium jointly with the Gairdner Foundation. So after 4 years of open, we have a robust ethical framework, we have a large collection, we have scientific collaborations, and we are basically including other groups, including the Douglas Brain Bank, that has one of the largest brain banks in the world. So the next, we're having some slowness with the slides, but okay. So then there's the Open Early Drug Discovery Unit. Essentially, it's using iPSCs, induced pluripotent stem cells technology, develop industry standard assays that are disease relevant, and use these to be able to work with industry and academic groups to try to identify what are the targets that are needed to be able to modify diseases. After 4 years in the open, we have a good collaboration with industry, philanthropy, and with the governments through grants. We have agreements with different entities, the Structural Genomics Consortium and ADMARI, collaborations with actually four pharma industries, including money, significant money from them, collaborations with open science drug development companies. And I should say that these collaborations with these companies are in the open, and so this is making the data available. The second aim of TOSI, of the Tannenbaum Open Science Institute, is basically to convince others to come on board. One is document what we've done and share. So the principles, the buy-in process, the toolkit for implementing open science, the open science contracts and documentation, ethics framework, so on and so forth. So all of this is freely available, and any informatics developments will also be all freely available. We are basically in communication and I guess in exchanging some education, some collaborations with governments, with funding agencies, with many different universities and institutions. International as well, a lot of activity in Europe and different parts of the Americas, including Brazil, Asia, and with some international groups. We have on November 11, 12, the Neuro Gairdner Open Science in Action Symposium that we are doing with the Gairdner. It'll be, I think, a great symposium, and I would encourage you all to come and listen. It's going to be virtual. So just a few last words. When we were doing this, we were told a couple of things, like, will implementing an open science policy restrain young researchers? So we were told this is going to be bad for young researchers. Completely wrong. Young researchers are very, very into open science. They believe in open science more than us older researchers. And we have had people who have decided to come to the Neuro for research, even because of open science. So that's not a problem. Will open science prevent collaboration with external stakeholders? No. In fact, we have more collaborations, including with some non-traditional collaborators. Will ethics be an issue? No. Robust ethical framework is in place. Patients, highly supportive. Patients feel empowered. This is very positive for patients. Will pharma see the value and embark? Yes, we have more pharma investment at the Neuro now than we have ever had before. And I guess I have to do twice to do once. Okay. Okay, sorry. Yes, will donors embark? Great support from philanthropy. We have a 400% increase in revenues from philanthropy. Philanthropists like this idea, and so do disease associations. They want acceleration of results. Will other academic institutions might be reluctant to join because of our position on IP? In fact, we have many institutions that I think will soon adopt open science like us, maybe not identical, but very much like us. And Stanford has created a new center for open and reproducible science. So open science will transform the way we all do science, accelerating discovery and maximizing impact. Thank you very much. Acknowledgements, the many, many students, postdocs, and technicians who have worked with me, my many colleagues and collaborators across Montreal, Canada, and the world, thousands of patients and families, and many funding agencies. So I couldn't name them all, there's just too many. Thank you.
Thank you, Guy, for a great talk, great science on neurogenetics, and a very challenging to all of us, challenging us to think about open science and how we can use it in our own research and in our own research institutes to change the way we develop new treatments for patients. So thank you very much indeed. And now we're going to move on to our next awardees, who are the Quarraisha and Salim Abdool Karim, who are this year's John Dirks Canada Gairdner Global Health awardees. So over to them.
Broadly in the field of HIV infection, and sub-Saharan Africa bears a disproportionate burden of the global number of infections. So it's home to about 70% of the global burden of infections. A unique feature and characteristics of this epidemic is that young women acquire infection 5 to 7 years before their male peers. There's a disconnect between what is available to prevent infection that women can use and where the burden of infection is. And that gap is on women-initiated prevention technologies that we have focused on.
We started off by describing the epidemiology of HIV infection in women. And so the entire body of knowledge that we're being recognized for comprises our work to find solutions to slow the HIV epidemic in young women. And they range from studies that have tried to answer the question as to why young girls have such high rates of HIV infection. And then we've gone on to testing a whole range of different products and eventually we showed that an antiretroviral drug, tenofovir, in a gel form was able to prevent HIV. Our research was the first to demonstrate that pre-exposure prophylaxis with antiretroviral drugs could prevent sexual transmission.
The direct impact is access to technologies that work. The indirect impact is really what's inspiring in terms of how the communities that have been devastated by this epidemic have kept their resilience.
So we hope that our research now into the future will be able to develop technologies that will be long-acting. So right now we are developing a technology which uses tenofovir in an implant that would last a whole year, that women can use comfortably, and that it would get widespread implementation to make a real impact on the HIV epidemic. It took us 18 years before we had a first positive result. That kind of perseverance is what's needed if you are to make major breakthroughs.
Greetings, everyone. I'd like to start by thanking the Gairdner Foundation for conferring the 2020 John Dirks Gairdner Global Health Award to Salim and myself. I also want to use this opportunity to congratulate all the 2020 Gairdner laureates, and to especially thank Janet and her team for organizing an amazing series of activities, even in the face of COVID-19 challenges, including this wonderful virtual event that enables lots of you to join us. I think we need to get the slides going. Okay, thanks. I'd like to use this opportunity to pay tribute to three of our mentors, Mervyn Sasser, Zena Stein, and Jerry Kuvadia, on whose shoulders our scientific career has risen and grown, and thank them for showing us the importance of science and social justice, and also in undertaking science in Africa, not to lose sight of excellence. I also want to use this opportunity to thank our parents, our siblings, our children for the years of support, and the many thousands of research participants, our large study teams, collaborators and funders, and everyone who has contributed to enable us to undertake this huge body of research that we'll share with you very shortly. Now, as you saw from the video preview, we will cover just over 3 decades of research. So I'll start by covering the first 2 decades, and Salim will follow with the next 2 decades, with the next decade rather. And I want to start by declaring that we have no conflicts of interest. So just a very quick overview. I'll be covering the issues and providing some context on the high HIV burden in young women in Africa. I'll specifically elaborate on the age disparity and the cycle of transmission that continues to sustain high incidence rates despite a high background prevalence of HIV infection. And then I'll talk about empowering women with a new prevention strategy. So just looking at the global epidemic at a glance, in 2019 globally, there were 38 million people living with HIV, just nearly 700,000 deaths, and 1.7 million new infections. And this is despite the fact that we have just over 25 million people on antiretroviral treatment, and where antiretroviral treatment has changed AIDS from an inevitably fatal condition to one that is chronic and manageable. So we've also made immense progress in reducing transmission from infected mothers and infants, where in Africa, for example, we had rates of 30 to 40%. Globally, we're looking at less than 2%, and in fact have set the target of elimination of vertical transmission. But let's return to the problem of new infections that continue to be a huge challenge. And we take that 1.7 million new infections. Another way to look at them, it's just over 4,500 new infections each day. Now, when we look at those infections, more than 70% of them continue to occur in sub-Saharan Africa. In addition, in sub-Saharan Africa, young women have about 1 in 4 new infections. Now, if we have to look at South Africa, it has less than 1% of the global burden of infection, yet it is home to about 8 million infections and ranks #1 in terms of the country with the highest burden of infection. Within South Africa, we have a diversity of epidemics with the highest rates of infection on the East Coast, and the province of KwaZulu-Natal, where we are based, is the epicenter, in fact, of the pandemic. And I want to take you to a rural community about 1.5 hours west of Durban, where we've been working for the past 2 decades. And I want to start by sharing some data with you from high schools, and particularly looking at learners in the first 2 years of being in high school. And what you see here in young boys, that by age 15, about 1% is infected with HIV. And up to about age 19, you see this less than 2%. In fact, when they reach age 20 and over, it still remains under 2%. I'm having a bit of a challenge multitasking here, but I think I'm getting there. So in contrast, if we look at young women, what we see already, if you compare young women age 15 to young men, they have about 3 times more infection. If you go to age 17, it's about 6 times more infection compared to young boys. And by age 19, 20, you're seeing almost a 10-fold difference between HIV infections in young women compared to young men. So I'm going to talk a little bit more about this age disparity and how important it is in terms of HIV transmission, particularly in Eastern and Southern Africa. So way back in 1990, we undertook one of the earliest community-based surveys. And here what we found was an overall prevalence of less than 1%. But when we did the age disaggregated analysis, we found, if you follow the red line, 4 times more infection in women compared to men. However, when we did the age sex disaggregated analysis, we found that up to age 10 to 14, HIV infection was rare in both young boys and young girls. But by age 15 to 19, while HIV infection remained rare in young boys, it was almost reaching peak prevalence in young women, and HIV peaked at age 20 to 24 in young women compared to 30 to 35 in young men. Subsequently, data started to emerge from across Africa, and what you see on the right is that in most countries across Africa, there's the strong age-sex difference where there's more infection in women compared to men. When we look at there were other studies looking at this age disparate relationships in Africa, and they came up with differing results challenging our observation of this age-sex difference and its importance. And it wasn't until 2015 when new technologies became available, and particularly phylogenetics, that we were able to study the directionality of transmission between young men and women at a population level. So using a population-based analysis, we were able to sequence over 90 clusters of clusters from about 1,500 sequenced viruses that were from people who had recently been infected, drawing from about 10,000 households. We were able to show quite unequivocally that this schematic, as captured in the schematic, that young women under 25 years were getting infected almost uniformly from men 25 to 40 years. In fact, an age difference of about 8 years was commonly linked with HIV acquisition in young women, an age difference of 8 years with a male partner. In turn, these young women, when they become when they enter the 25 to 40 year age group, are now having sex with men who are their peers, and you see bidirectional transmission taking place between women 25 to 40 and men 25 to 40. Of note is that 40% of these men 25 to 40 years simultaneously have relationships with young women. And it is this simultaneous relationships that foster the cycle of transmission and continue to perpetuate transmission of infection. And if we can stop infection in young women under 25, we can start to see some hope in breaking chains of transmission and achieving epidemic control. When we shared these data, and after it was published, we shared this initially with UNAIDS, and UNAIDS in 2016 dedicated their report, which is really the guidance for the rest of the world on what to prioritize, how to prioritize. And you can see here how important this evidence on the cycle of transmission was in shaping not only the response to the epidemic globally, but that this data then also came back to South Africa and was included in the South African National AIDS Council strategy for 2017 to 2025. And a key priority of that was based on the cycle of transmission and the importance of preventing HIV infection in young women. Now I'm going to move on to the next part around the gaps that we see. So I think at this point, I hope I have given you sufficient evidence to understand and appreciate the high burden of infection in young women. But what is now important to look at is what are the available prevention strategies. And at that point, when we first published this data in 1990, and indeed for nearly 15 to 20 years thereafter, the options that we had available was abstinence, behavior change, male-female condoms, and medical male circumcision. We ask ourselves, as many young women were, which of these are actually tools for young women in Africa? Indeed, in 1990, Zena Stein published this commentary in the American Journal of Public Health that highlighted this gap between the vulnerability of young women and the unavailability of technologies or tools that young women can use to prevent HIV infection and protect themselves without having to seek cooperation from their male partners. And you can imagine if you were a 20-year-old woman having a partner who's 25 years, we have few years of education, few economic opportunities, and dependent both economically and socially on a man in your life, to now start to negotiate with him about using things that require his cooperation. So since 1993, we and others have been undertaking microbicide trials, and microbicides are chemicals that women can insert into their genital tract that can potentially prevent HIV infection. Very early products, we're also looking at preventing other STIs and potentially contraceptives. And what we saw was, as the epidemic, and here using the data amongst antenatal clinics, seeing how the epidemic is growing, what we saw was study after study using the products that were available. We were constantly not finding the results that we wanted. In fact, what we became well known for, all of us working in the microbicide field, was experts at failing. In fact, preventing HIV infection was such a huge challenge that there were something like 37 randomized controlled trials being undertaken across the globe, and each time we were meeting a negative result. And so we continued and persisted. In 2003, as we started to provide antiretroviral treatment to the large numbers of people infected in Africa through support from the Global Fund for AIDS, TB, and Malaria, and the Presidential Emergency Program for AIDS Relief, we came across a drug called tenofovir. And tenofovir, we found, was an effective therapeutic agent. It had a good safety profile. It was rapidly absorbed, had a long half-life, and rapidly suppresses viral replication. And we had some evidence of its protective benefit in monkey studies. So in 2003, we said, let's try using an antiretroviral-based microbicide, and we initiated the CAPRISA 004 trial to assess the safety and effectiveness of tenofovir gel to prevent HIV infection. Now, many of the women, particularly in rural communities who don't see their partners every day, understand that they don't need a method that needs to be used daily, and needed something that they can use around the time they have sex with their partners, which could be anything from weekly, monthly, or 3-monthly. And we were inspired at that stage with some data from the mother-to-child transmission field, where they were giving 1 dose of nevirapine to the mother at onset of labor and a dose to the baby within 72 hours after delivery, and they were seeing a 40% reduction in transmission. So we developed this FAT24 coagulally related gel use, where we counseled women to insert 1 gel up to 12 hours before they were planning to have sex, another gel within 12 hours after sex, and not more than 2 gels within a 24-hour period. And this was because that was the safety data that we had available at that stage. Now, even as we initiated this trial, this commentary appeared in Nature Medicine declaring that this trial was doomed by design. And this was based on interviews from some of our peers and colleagues across particularly in the US. Notwithstanding, we continued with the trial, and in 2010, we announced the results of this trial at the International AIDS Conference in Vienna. And what we found was that in our trial, tenofovir gel prevented HIV infection in women, 39% overall, and it was high, up to 74% in women where we had high detectable levels of tenofovir. And Salim will continue in the details that he will provide to explain the importance of the threshold of drug levels on the efficacy of tenofovir gel in protection, and also some of the reasons behind these results that we see. But at this time that these results were announced, they were declared as one of the top 10 scientific breakthroughs by Science, and also had wide popular media coverage, and also regarded as a top breakthrough in the journal Lancet. And as I said earlier, it reflected the immense challenges all of us globally who are working on finding prevention technologies we're facing in terms of coming up with effective strategies to prevent HIV infection. These data, so if you think about it, in 2003, from the idea and several studies of the oral form of the drug being completed, led in 2015 to the guidelines being released by WHO in 2015, including ARV containing pre-exposure prophylaxis, also referred to PrEP, as part of our prevention strategy. It was adopted in South Africa in 2017, and we continue to contribute to improving and enhancing access to oral PrEP to young men and women who require it in addition to other technologies. What we also found in the CAPRISA 004 trial was that tenofovir gel prevented HSV-2 infection. Now, herpes simplex virus 2 infection is a very common sexually transmitted infection, and in fact is more common than HIV. And this was a serendipitous finding that we found. And what we also know is that there's a bi-directional relationship between HIV and HSV-2. So finding this was a wonderful surprise and an important contribution. I'm now going to hand over to Salim to walk us through the next decade of our research and to reflect on some of the underlying biology and factors contributing to PrEP efficacy and some of the new research that we currently have underway. Thank you, Salim.
Thank you very much, Quarraisha. It's a great pleasure and an honor to deliver this address as recipients of this year's Gairdner John Dirks Award. You've heard from Quarraisha that within the global HIV epidemic, the highest burden of HIV is in young women, and young women remain the highest priority for overall global control of this epidemic. And that when we look at why are young women having so much of HIV and why is the burden so high in this group, we find that age disparity plays a big role. That when we look at these young girls having sex with older men, about 8 to 10 years older, that they present this high risk to these young teenage girls. And in seeing this challenge of age, we began to understand that we have to find a way to empower young women. And Quarraisha's already shared with you the way in which we proceeded with developing tenofovir gel, testing it, and then eventually reporting the results of the tenofovir gel trial in Vienna in 2010. So what I'm going to do now is to take from that starting point and from that foundation, I'm going to tell you about why we had a problem with the efficacy of tenofovir gel. And I'm going to then share with you what are some of the underlying risk factors that we've discovered since our discovery of tenofovir gel, and what are we doing now? What lies ahead? And I'm going to end off with just giving you the 30-year timeline, because it's 30 years almost to the day when Quarraisha and I first started working on this problem. We identified this high risk in young women back in 1990. And we, as you heard from Quarraisha, we spent the first 18 years from 1993 to 2010 doing microbicide research and failing dismally, study after study after study. And so I'll share that timeline with you to give you a sense of all of the challenges that we dealt with as we went along. So I'm going to quickly tell you about now the 3 clinical trials that produced results of tenofovir gel. So you saw that the CAPRISA 004 trial produced a 39% protection. 2 other studies using the daily form of tenofovir gel showed lesser results. And that came as a big surprise. Why did they not show a similar result? And partly it was because of the daily regimen that we saw lower levels of adherence. That when women are not able to use the gel every day, that we see a much lower level of usage. When you reanalyze all 3 trials in a standard way using detectable drug levels, In other words, looking only at the women who have evidence of drug use, of the use of tenofovir gel, there we see a very consistent effect. A protection level between 52 and 57% across the 3 trials. So that told us we have a problem. We have a problem because women aren't able to use the gel when they really need to. And so that egged us on. We wanted to get a sense of, why is it that this adherence is proving to be such a big challenge? Well, it turns out that it's a big challenge because when we looked at what is the protective levels of drug that you need in the vagina, then we realized that you actually need pretty high levels of drug. And so it's having these high levels of drug are essential. And with the lower levels of adherence, when women were missing doses, those levels were going below target. And so when you look at this Kaplan-Meier graph, you get a sense of that in those women who had lower levels of tenofovir, levels below 1,000 nanograms per mil, they were very similar to placebo. So the blue line and the red line are very similar. Whereas the women who have high tenofovir levels above 1,000 nanograms per mil, they are in the green line, which is a much lower risk of HIV. And as we looked at this, we began to look at, so what might be influencing the levels of tenofovir in the vagina? And so we set up a collaboration with colleagues in Canada. And in this particular study, we showed that actually the vaginal microbiome plays quite a big role in that women who had lactobacilli dominance had a higher level of efficacy from tenofovir gel, and that women who did not have lactobacilli dominance had a much lower efficacy at 18%. Now, lactobacilli are the good organisms. They produce lactic acid, they reflect a healthy vagina, and they produce peroxide and many other things that help protect the vagina. Well, when we looked at this problem and realized that when we were looking at lactobacilli dominance and when we looked at a deficit of lactobacilli, we realized that other organisms, remember that the vagina has thousands of different organisms, and in the microbiome studies we have done, we have identified more than 5,000 different organisms. But one in particular, Gairdnerella vaginalis, played a key role. In women who had low levels of lactobacilli, they had higher levels of Gairdnerella vaginalis. And in having higher levels of Gairdnerella vaginalis, we realized that Gairdnerella was actually metabolizing tenofovir in the vagina. Then what we showed is that within 4 hours, the levels of tenofovir in the vagina were halved in the presence of Gairdnerella. But when you had lactobacilli dominance, your tenofovir levels remained high. So now we began to understand the importance of having adequate and therapeutic levels or protective levels of tenofovir, and we began to understand the role of the microbiome in influencing that. And that led us to an intermediate step because we didn't quite figure out how one goes to the other. And that's when we identified the role of genital inflammation. And genital inflammation is looking at inflammatory cytokines, little proteins. And when we found that the majority of these inflammatory cytokines were raised, then we found that we categorized those as women who have genital inflammation. Now, it's not a clinical condition because they don't have evidence of clinical inflammation. It's a subclinical inflammation. And basically what we show is that in this paper in Nature Medicine, we show that among women who have no inflammation and who have high levels of tenofovir adherence, in other words, they're taking the gel, we show very good protection, very high levels of protection. On the right-hand side, the black dotted line shows the very low risk of HIV among women with no inflammation but high levels of drug. Among women who have high levels of drug and inflammation, or who have low levels of drug, whether they have inflammation or not, we see very high levels of risk of HIV. Now that told us that we have to look at the three together, the levels of drug, the vaginal microbiome, and in particular the relationship between lactobacilli and Gardnerella, and then also the role of inflammation. Now we were trying to understand, how does inflammation impact on all of this? And that's when we started trying to better understand the role of inflammation. So in this study, this is a heat map. Each woman is reflected as a little square on top of the heat map. A blue square reflects a woman who became subsequently infected with HIV. A yellow square is a woman who remained HIV negative. And when we look at each woman is one column, and each row is a cytokine. Among women who have most of their cytokines raised, in other words, the red areas, we see most of those women went on to acquire HIV. In other words, most of them are blue squares. On the other side of the heat map, where most of the women where most of the cytokines are low or normal, there we see that most of those women didn't get HIV. So now we began to understand the role of genital inflammation and being a key risk factor. And in fact, it turned out to be a major risk factor, a biological risk factor for HIV acquisition in young women. And that risk factor showed a threefold increase in the risk of HIV in the presence of genital inflammation. So what causes this genital inflammation? And that became a challenge for us because we looked at it from multiple different angles. One of the factors we found played a role was a bacterium called Prevotella. And one particular Prevotella bivia, was often associated with high inflammation. But Prevotella bivia is not very common in women, so it couldn't really explain a very high proportion. However, human papillomavirus is much more common. And so we decided to look at human papillomavirus and get some idea as to the role of human papillomavirus in causing genital inflammation. And if we look at women who are HPV negative, in other words, who do not have human papillomavirus infection, either acutely or persistently or clearing it, that's the women in the blue line at the bottom, showing their very low risk of HIV infection. Among women who are acquiring HPV, or who had a persistent form of HPV, or who are clearing HPV, those are the other three lines, and they all have high risk of HIV. Overall, we show a 2.5-fold increased risk of HIV in women who have human papillomavirus infection. So now we have a clear understanding of genital inflammation in the vagina playing an important role. The HPV is associated with that. So how does genital inflammation lead to and increase the risk of HIV? And in this analysis, we show that these increased levels of inflammatory cytokines in the genital tract, they increased the HIV susceptible target cells. Now, HIV can only enter into cells that have the CD4 receptor. Now, these cells are increased in the presence of genital inflammation. Not only are there more cells, but these cells rise up and become very close to the surface of the mucosa, making it much easier to infect. So now we began to unravel the equation, understanding the role of the microbiome, the role of HPV, leading to genital inflammation mobilizing CD4 cells, increasing the number of target cells, making it easier for women to acquire HIV. Understanding that now gave us an opportunity to see what could we do to improve on the technologies we had already worked on. And so I'm going to share with you some of these novel technologies. And why are we still working on this 30 years later? We're working on it 30 years later because it remains to this day, as it was 30 years ago, one of the main causes of burden of HIV. 30 years ago, women had contributed the highest burden of HIV. To this day, women in Africa, young women in Africa, remain the highest burden. So while we have made great progress in HIV treatment, unfortunately, we have not made as good progress in prevention. We are lagging far behind. Globally, we had set some 7 years ago the target of 500,000 new infections in 2020. Last year, it's estimated by UNAIDS that there were 1.7 million new infections. We are way off target. And we're never going to get to that target unless we can find some way to protect young women. And so that's why we persist in this challenge. Now, there are many technologies being studied, all kinds of different approaches, trying to alter the microbiome, looking at multi-purpose technologies, developing injectables. So the field is involved in a multitude of different options that are being investigated. We have chosen to focus on two things as long-acting formulations, trying to get away from the need to use a gel every day or take a tablet every day, and rather to focus on how can we provide a technology that women will simply have and not have to think about it until they come for their next dose. And so we're looking at passive immunization with antibodies and a long-acting implant as another option. So let me talk briefly about the long-acting implant. We're collaborating with our colleagues in Los Angeles, and together with them, we're making an implant. This implant has a much more potent form of tenofovir, a drug called tenofovir alafenamide. We have been able to put about 110 milligrams of tenofovir alafenamide into an implant. It's about a matchstick size, and it's inserted under the skin in the arm, just like the way a contraceptive implant is inserted. And this implant, we anticipate, will provide adequate protection for a year. Now, we don't know if it works yet. That's what we currently study. But if it works, and if we can deliver enough tenofovir through this implant to protect a woman over an entire year of exposure, it's a game changer because it will enable us to protect whole groups of high-risk young women with this technology. So watch this space. We are now at the tail end of the phase 1 study, and we'll be going into phase 2 trials soon. You know, it's a long road. We've been working on the implant now for almost 7 years. It's going to take us another 3 to 4 years before we have results. So it's a long road to get there. We're also working on another technology, and this is a broadly neutralizing antibodies. Now, when any person becomes infected with HIV, the body naturally produces antibodies. But those antibodies are virus-specific, or what we say strain-specific antibodies. The antibodies they develop will only kill that strain of HIV. But in 2010, 2 groups discovered that there are a small proportion of people who can develop a broadly neutralizing antibody. Their antibodies can kill viruses that have a very wide range. They can kill viruses from pretty much all over the world. And so we decided to go and look for in our patients to see if any of them had these antibodies, and we found 8. One in particular, known as CAPRISA 256, her antibody was special. Firstly, we tested it against 200 viruses from all over the world, from every continent, all of the different countries. And what we found is that her antibody could kill 4 out of 5 of those viruses. Now, that was striking to us. But what was also interesting was it was highly potent. So we decided we've got to pursue this. So we sent a student to go and work on this antibody at the Vaccine Research Center at the NIH. And working with them, she was able to identify that one cell that makes this antibody. We were able to clone that cell and manufacture the first batches of this antibody. We then put those into animal studies. And we sent it to our colleague, Dan Baruch at Harvard, and he injected monkeys with this antibody. So the 9 monkeys in the left-hand side diagram, the 9 monkeys that received the placebo, it's a blinded trial, all of them become infected with the monkey form of HIV. In the 3 diagrams to the right, looking at differing doses of CAP256 antibody, none of the 12 monkeys that received the antibody became infected. So armed with these exciting results, it protects monkeys. They don't get infected if they've got this antibody. We then proceeded to make human quality antibodies. And working with the NIH, we now have completed manufacture of GMP lots of this antibody, and we've started injecting them into humans. And we currently just completed the first in human trials of this antibody, and we're now proceeding into the phase 1 trials. So very exciting for us to see how we started, with an antibody that we found in a woman, all the way now to where her antibody from CAP256, who still comes to our clinic, we see her all the time, she's one of our patients, and that her antibody may be able to protect whole generations of young women in Africa. So let me end off with the 30-year timeline. So we started off in 1990 with this initial study that showed the disproportionate burden in young women. We saw Zina Stein's article and all of the studies that we undertook over the years, and they are all shown in the blue squares. And then the gray squares under the line, these are other studies that were being done in the same field by our colleagues. And in looking at all of this, you get a sense of this 20-year timeline from 1990 all the way to 2010, when we announced for the first time a positive result from a microbicide. And that since then, as we have worked on these microbicides, we have found that in the last 10 years, we have now focused on trying to improve it. A range of different studies looking at rings, looking at injectables, and looking at now the TAF implant and the broadly neutralizing antibodies. And at the bottom of the graph, I'm showing you all of the basic science studies that have helped us unravel why these women are at high risk. So let me conclude by simply pointing out that reducing high rates of HIV in young women in Africa is key to controlling the global epidemic. That age disparity in the partnering patterns is critical. Genital inflammation is a major biological factor. We need longer acting strategies. We are developing 2 such strategies, and that up to today, it remains a high priority to find a new HIV prevention tool for women. I'd like to end by acknowledging the many, many investigators. We have a team that now is well over 200 senior scientists, and I can't even count the number of postdocs and students, and to all of our funders that made this possible, a very big thank you. This recognition is to all of their contributions. Thank you very much from Quarraisha and I.
Well, thank you very much, Salim and Quarraisha. That was a great talk, highly inspirational to see how, over a 30-year timeline, by sticking with it and following your compassion for the women, to really come forward with treatments and preventions, and very exciting potential new studies as well. So congratulations. So now we're going to move on to the last of our lectures for this year, and this is the final of our 5 Canada Gairdner International Awardees, with Roel Nusse from Stanford University in California.
At how organisms develop, starting from a single cell, dividing and then generating all kinds of specialized tissues. One major question that comes up is, how is that actually being controlled? And what drives cells to proliferate? in certain instances and what makes them to become specialized in other events. There is a major role for how cells talk to each other during development. The challenge that we set out originally, which is now many years ago, was to understand how breast cancer in the mouse would be caused by a tumor virus, a retrovirus. We found that there was a gene that was present in the normal cells that became activated as a consequence of the retrovirus. What emerged from all of those studies is that there is a tight connection between how cells normally become regulated to proliferate, to divide, and the way that cells divide in an abnormal way, which happens in cancer. And that all of that together has brought that gene, which we now called WNT, W-N-T, together in a framework where the regulation of that gene itself was very important to study, to understand what happens in normal development, but also when the gene becomes misregulated, how cancer arises. Is it possible in the future to use WNT, which is a major signal to promote the growth of tissues and cells, to use it in a clinical setting? Can we use it to promote the healing of tissues? Is it possible to interfere with WNT signaling in forms of human cancer? And there are now many attempts going on to regulate the WNT pathway to inhibit the growth of cancer cells. And those efforts are now worldwide. I received a phone call from Janet Rossand, the president of the Gairdner Foundation. I knew about the prize. I was not aware of the fact that I was being nominated or considered. The importance of an award comes from the people who got that award before you. And if you go down the list of previous Gairdner Award winners, it is a very impressive list. There's many scientists who at the time and later also made major discoveries. So let me start by thanking the Gairdner organization for this very significant honor. I was very pleased, as also mentioned in the introductory talk, by getting a call. And I will talk today about some of our past research on WNT signaling, but also what we are doing at the moment. This, of course, is a virtual talk. In preparing my presentation, I was contemplating what it would be like to visit the site of the ceremony, the gala, and I discovered the building, the Ontario in Toronto, is quite interesting. It seems a very mixed architecture, an old part and then a new roof as well. So WNTs are now known to be signals that are acting between cells that are in close proximity. They're signals, but the WNTs are most widely used signaling mechanisms between cells, and that comes out of many different corners of research, but also from considering how animals have evolved. And in this slide, I want to highlight that over the course of evolution, animals, including porifera, these are the sponges, cnidaria, ecdysozoa, and chordates, including ourselves, have become highly specialized. They contain many different cells, and cell division is quite tightly regulated. Now, that all has evolved from unicellular organisms, where that subdivision of certain cells dividing and others do not, has not evolved yet. Now, when you look at the early stages of development of these animals, and this is true no matter what kind of animal you're looking at, sponges or chordates, you see that the early embryos evolve from clumps of cells where initially all of the cells are undifferentiated. And then what emerges is that there is a local growth zone. So certain cells in the embryo, they start to divide at a higher rate, and others do not. And as you can imagine, what happens then is you get an elongation of a ball of cell into something that looks more like having an axis of asymmetry and an anterior and a posterior domain in the developing embryo. The anterior domain then evolves into the head of the embryo and the posterior domain in the tail. So that local growth is very important to set up asymmetry in a developing embryo. Now, quite strikingly, if you now start to compare...
I found this on the web for port to shut up asymmetry in a developing embryo. Check it out.
Sorry about that. If you now start to compare the expression of wind signals in the developing embryo, and this again holds true for sponges all the way to the chordates, you see that early on, winds are expressed at exactly that posterior growth zone. And other work has shown that if you remove the winds from these embryos, you wouldn't see that early proliferation of cells. So winds are essential for cell proliferation and setting up the primary body axis of all these branches of animals. And quite strikingly, if you now start to look also at the origin of the winds in terms of evolution, you see that these unicellular organisms, where everybody divides and there's no real difference between the individuals, they don't have winds. But as soon as you now get the evolution of the metazoa and the branches of the metazoan kingdom, you see the evolution and the origin of the wind genes as well. So that illustrates that winds are important for proliferation, but in a regulated way. Now the same is true if you start to look at tissues in adult organisms and the way that those tissues grow. Invariably, tissues grow from progenitor cells, and those progenitor cells are instructed by signals that are acting in a very local way to tell the progenitor cells to divide and then to make their differentiated daughter cells. Those differentiated daughter cells are commonly not dividing, they're specialized, they execute the function of the tissue. And when those cells are dying or removed from the tissue in another way, it is only from the progenitor cells that are actively dividing that you get the maintenance of the tissue. Again, all under the control of these local signals. The local signals in many different tissues are, again, quite often members of the wind gene family. They are acting very locally, and they make sure that it is only the progenitors that are retaining the proliferative capacity, and that as soon as the daughter cells of the progenitor cells are now moving away from the wind source, they now acquire this state of being differentiated and not proliferative anymore. In addition, in many tissues, the differentiated cells are actually arrested in the cell cycle through the expression of active inhibitors. And this is now quite fundamental, not only in understanding how tissues are built up and how they're being maintained, but it is also very fundamental in considering the likelihood that certain cells may turn into a cancer cell. It is now clear that many of the cancers are actually arising from the progenitor cells, and that makes sense because these are the cells that have retained their proliferative capacity. And at the same time, the differentiated cells arrested in the cell cycle are less likely to become cancerous. Now, what have we understood in the course of the years about wind cycling? I'm always displaying wind cycling in a state where there is a cell that is not activated yet by a wind. On the surface of those cells are receptors, LRP and frizzled, and then in the cytoplasm is a protein called beta-catenin. And the beta-catenin molecule is kept in an off state. It is inactivated by the interaction with these other molecules, including a protein kinase. Now, in the other state, when a Wnt protein comes along, again made by a neighboring cell, and then binding to the receptors LRP and frizzled, you get a rearrangement of this complex in cells, and that results in the beta-catenin protein now going to the nucleus, and they are binding to a TCF molecule, and that now results in the activation of target genes of the Wnt pathway. The cell is now altered with respect to its state. It is making a whole bunch of new genes and new proteins, and among those genes are genes like axin-2 and LGR5, and another gene that I'm going to tell you more about, and that's called TBX3. LGR5 is a molecule that has received a lot of attention as well. It is also a receptor, just like frizzled, but it doesn't bind to Wnt itself. It binds to another protein called R-spondin. Now, the knowledge of Wnt sickling for many years was initially coming from genetic experiments. And included in those genetic experiments was an experiment that I did, resulting in a paper that I published together with Harold Farmers, my mentor at the time, in 1982, where we were studying how the mouse memory tumor virus and retrovirus would cause breast cancer in the mouse. And what we found is that by a process called insertional mutagenesis, the proviral copy of the mouse memory tumor virus was landing next to a gene, and we cloned that gene by virtue of the fact that it was sitting next to that retroviral proviral DNA. And at the time we called that gene INT1, and later we renamed it into Wnt1. So this was the first member of the Wnt gene family that was molecularly cloned, and then subsequent work had led indeed to the realization that Wnt are very potent factors inducing cancer, for example, memory tumors in the mouse, but also cancers in other contexts. So because of the genetic discovery of Wnt and also the components of the Wnt pathway, it was very important that we got a better view of how these Wnt molecules would actually work. And therefore it was very important that Carl Willard, a postdoc in my lab, purified the Wnt protein in 2003, and thereby showed that the Wnt protein is actually modified in a very unusual way. It is modified by a lipid, a palmitoylate, and it sticks out, as was shown by work done by Claudia Janda and Chris Garcia at Stanford. It sticks out of the Wnt protein. And that now makes it possible that the Wnt protein first binds to the receptors, the frizzled, using that lipid, but also by sticking the lipid into a membrane, binds the entire Wnt molecule to membranes. And it implies that Wnt signals are indeed acting more or less as a membrane-bound protein, acting over a very short range in all of the contexts where these Wnt molecules are active. So that purification of Wnt and the subsequent identification of the R-spondin molecule has led now to the possibility to grow cells in culture from many different organs and so-called organoids. People have now been able to propagate cells in culture as organoids almost in the body, and they're all listed here, by virtue of the fact that Wnt sickling can maintain stem cells, but also lead to the differentiation of the stem cells in culture into an organoid. And it allows now to do many more experiments understanding how organs are built and how organs can actually turn into cancer cells, doing this all now in a cell culture system. It's also those identifying target genes of the Wnt pathway and how Wnt sickling actually works has led to the development of tools that allow you now to understand how Wnt sickling actually works in vivo in the context of an intact animal. And what was done by various labs is to build these reporter mice or lineage tracing mice, where we take the Wnt target genes like axin-2 and LGR5, and then allow those then to lead to the activation of reporters like LAC-Z or GFP in various different contexts. And that work has led now to the realization that you can actually use those reporters to label stem cells in vivo and to follow what those stem cells are doing, to generate clones of not only the stem cell or the differentiated cell, and thereby to identify stem and progenitor cells in many different tissues. And on the next slide, I have a list of the tissue and stem cells that have been identified over the years. This is reviewed in a paper that Hans Clevers and Kai Lowe and myself published in Science some years ago, where you can see that stem cells, for example, in the intestine, the mammary gland, stomach, etc., can all be labeled in such a way that you can identify where the stem cell is located, but also that the stem cell is going to generate these beautiful clones. For example, here in the skin, you see a clone of cells where you see the upper layer tenocytes being labeled as the protein of the stem cells in the basal layer of the tissue. And likewise in the intestine, mammary gland and brain, you can label Wnt-activated tissue stem or progenitor cells in a very robust way. And that is work that has been going on and has now been applied to many other tissues as well. Now, all in all, what we now understand is that Wnt signaling is very important to cause cells to divide. But how does it actually work? And if you think about the signal and the cell cycle and how cells are instructed by a signal to enter the cell cycle, people have found that most growth factors are actually activating S phase, when the cells start to make DNA and subsequently will enter then mitosis. What we found, however, in more recent work is that Wnt is not so much important maybe for entering S phase, but more for the control over cell division, over mitosis. And that came out of work that we have been doing more recently in the liver. In the liver, the tissue is subdivided into these big liver lobes, and then the liver lobes contain units of cells called liver lobules, where the hepatocytes in the tissue are organized surrounding a blood vessel. That blood vessel is called the central vein. There's other blood vessels called the portal vein. The central vein is a main organizing structure in building up these liver lobules. And what we found is that in the liver, one of the Wnt target genes, axin 2, is selectively expressed in hepatocytes that are just adjacent to that central vein. It is illustrated here by the blue staining in the tissue. And in the diagram here, you see that these pericentral hepatocytes, just abutting the central vein, they stain blue. And therefore, we suspected that they're activated by Wnt. And indeed, if you follow where Wnt itself is being made, we found by in situ hybridization that one of the Wnt family members, called Wnt9b, is made by the endothelial cells of the central vein. So here's a typical situation in a tissue. Some cells, in this case the endothelial cells of a blood vessel, they make Wnt. The cells that are immediately adjacent, in this case hepatocyte, they become activated by the Wnt and they turn on a Wnt reporter. So it is signaling again that occurs between cells that are nearby, but not so much to the cells that are more remotely present in the tissue. So we went on to do a whole bunch of experiments to characterize those cells and how they would behave under the influence of the Wnt signal. And what we found is that there is another Wnt target gene called TBX3 that was also expressed, just like axin 2, in these pericentral hepatocytes. Again, an in situ hybridization showing beautiful expression of the TBX3 RNA in the cells adjacent to the blood vessel, but not so much in the cells beyond the first layer. Now, this was very important for our work because it was known from work done by others that TBX3 is actually required for embryonic liver development. If you remove TBX3 from a mouse, there's virtually no development of the liver. So we then deleted the TBX3 gene from the postnatal liver as well. And what we found, this is work done by Tenny and Una in the lab, that it actually also blocks mitosis. So TBX3 is an important gene for translating the Wnt activity to a cell and instructing the cell to divide, because if there's no TBX3, even though there will be Wnt, the cells will not divide. Now you are in a situation that is typical for research. We have a signal, a Wnt. We found a target gene that is important for the signal, TBX3, and then you find your way downstream of TBX3. What is TBX3 now doing, and how does it interfere or act on a cell in terms of cell proliferation? Well, Una in the lab found that by chromatin IP experiments, that TBX3 is actually a DNA binding protein that is sitting next to the promoters of two other genes called E2F7 and E2F8. So that was known that E2F7 and E2F8 are also involved in the cell cycle, but they are actually known to be inhibitors of mitosis. So when you have E2F7 and E2F8 active in a cell may go through S phase, but that cell will not go through mitosis. Mitosis is blocked. And therefore the cell is now ending up with additional copies of its DNA, but it is not divided. And now you can probably see where this work is going. I told you earlier that we had TBX3 as a Wnt target, and that TBX3 was a repressor. And what we found indeed is that by binding of the TBX3 protein to the promoters of E2F7 and E2F8, the activity of E2F7 and E2F8 is blocked where there's Wnt signaling, and that allows now the cells to go through mitosis. So where there's Wnt and where TBX3 is active, cells can actually go through mitosis and actually generate two daughter cells. And that is now illustrated again in the slide that I see before. We had that subdivision of cells in a tissue where there is a population of progenitor cells. Those progenitor cells are able to divide and maintain that ability over the long term. They generate more differentiated daughter cells that now become arrested in the cell cycle. And in the liver, but also in other tissues, that arrest could be caused by the expression of E2F7 and E2F8. But in the progenitor cells, the expression of E2F7 and E2F8 is blocked. And it is blocked by that very local Wnt signaling, now through the protein TBX3. And the general message of that work is that Wnt turns out to be permissive for mitosis. Where it is active, cells are allowed to go through the cell cycle. Now, there are various links to cancer from this work. This is that pathway again, but what has emerged over the years, and that is with many different groups and many different laboratories, is that there are mutations in maybe all of the components of the Wnt pathway that can lead to cancer. There are mutations in AXIN and APC, but also mutations in beta-catenin that can lead to forms of cancer like colon cancer, liver cancer, and many other forms of cancer. Also, what has been found is that the TBX3 gene, as you can expect based on the way that it works, when it is active, it can actually promote cancer. And we have some data now that also in liver cancer, that TBX3 is one of the drivers of early liver cancer. But as you can also imagine, that E2F7 and E2F8, which are now opposing the act of the Wnt pathway, can actually block cancer and can actually act as a tumor suppressor gene. And then on top of that, the Wnt itself can be activated in mouse breast cancer. And that goes back, of course, to our early work on the link between Wnt signaling and breast cancer in a mouse. Now, the work brings up a number of questions. How general is all of this? Is that axis between Wnt and TBX, is that conserved? And does it play out in other tissues as well? And what we found, and this is work that we're still working on, of course, is that TBX3 and other T-box genes are often expressed in these growth zones that are so important for early embryos and also for the maintenance of tissues. And they are expressed often there together with a Wnt. I should also mention that E2F7 and 8 are widely expressed in tissues, but in the differentiated cells, and in those cells they can also lead to mitotic arrest. And one example, and I'm mentioning this because it is close to the heart of genital sand is that E2F7 and 8 are also required for trophoblast giant cells. Now in the final couple of minutes, I want to play out that link between Wnt signaling and the T-box genes. We're quite fascinated by the possibility that not only TBX3 would be activated by Wnt to maintain the proliferative capacity of cells, but also possible members of other members of the T-box family. And the earliest member of the T-box family that was ever identified is a gene called brachyury. It was known to be required for early mouse development, but also for the development of many other animals, including non-vertebrates. And let me now show you again that diagram that I started with, is that we have these branches of the animal kingdom, Cnidaria, etc., chordates, totally fascinating, of course, to understand where they all came from, and the local expression of Wnt that leads to the local proliferation of cells in these animals, no matter what their origin is. It turns out that very similar observations can be made by the expression of that brachyury gene. It has been identified that if you look at the early development of these Cnidaria, and again these are sea anemones, you see that the brachyury gene is expressed very locally at the site in the embryo that coincides with where Wnt are expressed. And likewise, if you start to look at the early mouse embryo, you see a very local domain of that brachyury gene, again a TBX3 family member, at the site where Wnt are expressed, and very important for the development of the growing tail bud. And even more so, it is now known that Wnt activity is necessary for the expression of brachyury at this local growth zone. So it is possible indeed that this whole link between Wnt signaling and the expression of a T-box gene, TBX3 or brachyury, is conserved all the way across the animal kingdom and is important for the development of the early axis of animals independent of their origin. Now finally, I want to acknowledge many people who have been contributing to my research over the years. And of course, we're living now in the time of Zoom meetings, so I took some screenshots of recent lab meetings to make these acknowledgements. And let me first show you that this is my current lab. I think that many of them are listening to my talk at the moment, but without mentioning their name, I want to acknowledge their fantastic contributions to our recent research. It also happened that earlier in this year, we had a reason to celebrate also with the past lab members, and I took screenshots of our Zoom meetings with many of my past co-workers in the lab. I also want to acknowledge with another Zoom screenshot my mentor and current friend Harold Varmus, with whom we had a meeting. I'm also very indebted to the family and my friends from Europe and also here in the United States. And finally, I want to acknowledge, of course, the support of my inner family, including my three sons, Ben, Jamie, and IJsbrand. IJsbrand and his wife, Jessica, recently had a baby. And interestingly, the baby was born in Canada and has the Canadian citizenship. And finally, I want to acknowledge the support over the years of my wife, Betsy. Thank you for your attention.
Thank you so much, Roel. That was a great talk and a great way to end 2 days of wonderful lectures. The Wnt signaling pathway is really fundamental to the evolution of the animal kingdom, and it's wonderful to see the conservation. And thanks for the trophoblast giant cells. I was going to ask you whether TBX and that pathway is obviously involved in polyploidy in the liver as well. We can talk later. At this point, that is the end of the laureate lectures for this year. The lecturers themselves today are going to be taking a break now, but then they're going to be meeting virtually with students in interactive sessions. We always like to do that, usually in person, but it's another Zoom meeting coming up soon. So thanks to our laureates for taking that on. It's very important outreach to the students, and I think you'll enjoy it. So today's program has been recorded and it will be posted soon on our YouTube channel at Canada Gairdner Awards. So please also check our channel for recording of other events, including yesterday's Laureate Lectures and Wednesday's Canada Gairdner Global Perspective panel, which featured the Abdool Karims, Dr. Tony Fauci, Dr. Soumya Swaminathan, and many other global health experts in a really exciting discussion talking about the pandemic impact on global health and how to be prepared to build forward better. I'd like to thank our partners, the National Research Council, CIHR, and the University of Toronto for their support. And I'd like to thank our Canada Gairdner Laureates for 2020 for their participation today, and in fact for doing the Canada National Lecture Week across the country. It may have been virtual, but they have been at many different universities across the country. And in fact, we haven't finished. There's a couple of remaining visits to UBC and Toronto next week. And I'd like to thank you, our audience, for your participation all week long. Please stay in touch with us at Gairdner through the year on Twitter, Instagram, and Facebook at Gairdner Awards, and check our website, gairdner.org, for updates on news and events. Thank you for watching.